right, we're live. I'll just say a word as introduction. Hello, everybody. Today is Monday, November 10th, 2025. It is Bioinformatics time. And today is one of these classes where I will talk very little, instead let you talk. We had a class project that we worked on, namely a hybrid genome assembly on Illumina and Oxford nanopore data. And as I understand it, you tested out different assembly strategies, both regarding filtering of the reads and then which settings for the actual assembly process. And we would like to share these results now. So ideally, we go from one person to the next, and you guys could, here on the screen through Zoom or on the whiteboard if you want to draw something down, if everybody could just speak 2-3 minutes about their project, little information. We had subdivided you guys into groups, so it might be a good idea if we do this group by group, that, like, for example, the group members speak first, if the same group, and then we go to the next group members and then to the next group. All right, any questions? If not, I understand, Bryce, you volunteered to start. Please tell us, you have 3 to 4 minutes, or 5 minutes if you wish. What did you do during the class project? What were the results? And if they didn't quite work out the way you wanted, what conclusions did you draw from it?
I didn't really change the assembling process at all. I just used unicycler, the Bactopia.
Can you share your screen?
Okay. Well, I'm not really going to be sharing anything. I can show you my submission, like what I used.
Sure. Yeah. Just so that we get it, and also the viewers of this video have a little bit of an idea of what you were doing.
Okay. So as you can see in my SLARM file, I really didn't change anything for the unicycler. I basically just did the unicycler very standard to what we had before. Tried to keep that as simple as possible. I didn't want that to skew what I had done. What I really differentiated with was the filtering. So what we did before is we did this keep percent at 75, and that means you keep 75% of the data basically. I don't know really how fitlong does it, but it takes the best 75%, or in this case 70% of your data, and then it does that. Fitlong has multiple options, I learned, for filtering your data. This is just the most common one used usually, but I also wanted to try the, this one I found interesting was the minimum quality. So if it had a quality lower than 8, it just thrown it out. Even if that was 98% of the quality, it was going to throw it all out. It had to keep above 8 of the quality score of 8. That's 8 out of 10. And then I also did a minimum length of 1,000. I didn't figure that would do that much, honestly, but I thought it was interesting, so I decided to throw that one in too. I found at the very end that the contigs were very similar to the standard. It meant that our quality, I think it meant that our quality before the filtering was even really, really high, above this quality score fitlong determined of 8. So this filtering that we actually didn't really do that much, from my understanding. Like we did, the contig sizes were very similar. I had 3 contigs, most likely the plastid, the full big one, and then also a virus. It was very normal. I didn't lose very much data really. I think it just showed that the filtering process wasn't really as necessary as I thought it was going to be.
Good point. Thanks for that. This is so you evaluated specifically the filtering process. Feel free, like I think we have enough time that everybody, or that for every person there is a quick question. Does somebody have a question? Maybe if that's all right, I would actually take the first question if that's okay with you, and then I'll let you do the questions, because this one is just so characteristic. The fitlong that you tested, that filters what?
It filters the like the short read thing, is my understanding, or the long.
Exactly, it's the long read. It filters the Oxford nanopore reads. Okay. And here's an opinion. I'm not really sure. I haven't tested this out, but I think the reason it's totally good that you tested different settings for that filtering process. But you then used unicycler, which primarily operates on the short reads and uses the long reads just for polishing. So I believe the reason why your
It
final
didn't
genome
change.
assembly didn't really change much is that it would have changed quite a bit if you had filtered the short
Short.
reads differently. The long reads, which are used for polishing anyways, or which are used primarily for polishing, to filter those more or less strictly, probably didn't
I
change
guess
much.
that's good to know as well too. Then
Right.
that it doesn't rely on long reads very much at all.
Well, under those conditions, if you had changed the filter long conditions with Dragonfly, the assembly that primarily works on the long reads and uses the short ones for polishing, there I suspect you might have seen larger changes. But it's still a valid comparison. Totally okay. Mm-hmm.
I believe
All right.
Weeza did something similar to me, maybe a little bit modified. But
Yeah, who else was in the group? And maybe we just go within the group. We'll just hear. Thank you for that. Could you stop the sharing on your side, and then Luisa can continue her sharing.
Okay.
All right.
Okay. So
If you just make it a little bit bigger, because it's a bit difficult to see. Just a tiny little bit bigger, if possible.
okay.
Mm-hmm. I think that works well.
Okay. So for me, I made changes to the filtering step. For the fit long, I prioritized long reads. So I wanted a minimum length of 2,000 base long, and then the minimum mean quality of 10. And then target base, I wanted to go with a coverage of 100 times coverage, so to speak. So I took a little bit overkill, but 500 million bases for that. And then I still was prioritizing length over minimum quality. So the length quality was 10, and the minimum was 5. I don't know how to explain. Yeah.
Mhm.
So that was for the nanopore reads. But for the Illumina, I just made changes to the leading and the trailing. So for every in the beginning of the reads,
Mm-hmm.
yes, sorry. If the bases have a minimum quality of 5, discard. If it has a minimum quality of 5 at the end, discard it. And in the sliding window, I picked 5 over 20. So for every 5 bases, check the minimum quality. If it's 20, cut it off.
So you were
onwards.
more strict.
Yeah, more strict. Yeah.
You were stricter compared
Yes.
to what we had as a
Yes.
default. Mm-hmm. And what were your results then?
I did have 3 contigs with the bacterial genome about 4.3 megabases,
Mhm.
and I also had that for the plasmid and the phage as well. I'm trying to find the contigs, but those are the. I still did have 3 contigs,
Mhm.
and I compared it with someone that a colleague that did not make much edits to the filtering. It was very similar. So
Okay.
I do support what Bryce said. The filtering did more or less did not do much, because even if you did it stricter, you still have very similar
Mhm.
results as well.
Did at least the time change a little bit? Because with the stricter filter, I would assume that you had less input reads.
Yes.
Did
so
that run faster at the very least, or was the runtime still very similar to
it did
to the
run for
default
7
conditions?
hours.
7 hours?
Yeah.
That's pretty much the standard. Well, it depends on how many CPUs you use, but
8.
yeah, I see that. Yeah.
Sorry, yes, I used 8.
All right, so more evidence to that the initial quality filtering does not do too much of a change. Right? Who else was in your group?
Okay, so first of all, I agree with what they said, because they shared, I mean, I don't know for Bryce, but Louisa shared her results, so we
Mhm.
got to see, and then it mostly had to do with playing around the filtering, just
Mhm.
tweaking things around to see if we would see any valuable change. But like you said, it wasn't any different. For me, I tried
Can
running.
you stop the share
Oh,
so
sorry.
that Kwesi
Okay.
can share a little bit?
right from is it sharing?
It will start in a moment.
So right from last week, I did some initial runs, but then they all kept failing. I even had 2 of them that ran for like 12 hours, and then they still failed. And so I tried looking up to see why it failed. And then this was
when you said it failed, you're talking about the Bactopia, the assembly,
Yeah.
right? The assembly, at some point, the assembly stopped, and then you probably tried to figure out a little bit why it stopped,
Yeah,
like
so
why the run failed.
yeah, so I just did cuts of the slam output, and then I figured out that from the beginning I was using the or instead of I was using the Illumina or instead of the filter. I don't know if you understand what I'm saying.
Like he used like
I was using
instead
the
of the filtered one. He used
the
like
paired instead of using the paired one.
Oh,
Yeah, so I corrected that and then I repeated the run. And then it still failed. So I did another one. This is I want to show you my slam script. So that
sure, yeah, please.
So I don't know why this is showing here, but it's probably because I deleted the.
Yeah, those are just
instructions
the
that were- Yeah, but
green
this
is
is
just
the-
extra white spaces.
This is the slam scripts that I submitted.
And
And this time around, as you can see, I used the filtered one, but then it still failed, and I really don't understand why it failed. So I think I came to your office, and then
yeah.
you said it's okay for it to fail, but we should.
Yeah, of course,
figure it
yeah.
out.
Like you don't have to have like the picture perfect assembly. From that, it looks pretty. That looks pretty good to me. Anybody see any major errors in that Slurm file here?
When I was doing mine, I had to put an absolute path for the nanobore, so I had to go through all the files it was through. I don't know if that did anything, because mine would fail if I just left it like that too. I had to put it through every single folder. it was
I
in.
did that too. I had an output file designated for mine. Mine had like it's going to here afterwards, and that's how it worked for me. And it worked first try, honestly. So maybe that output file is required.
I didn't have an output
Yeah, I
file
didn't.
required.
Okay.
Output file. Are you talking about a Slurm output file or an output file regarding Bactopia?
I had it in the.
You're talking about the elementary side, right? Yeah. I think you're talking about this side.
Just using yeah, for Bactopia, like
In this.
I use mine as like it was like /home and then like my username, and then I had to use like the get and the fit log, and then like whatever the file name was.
No.
Like I couldn't just put like /nanopore.
Yeah, I also used.
Like I had to actually like go
absolute
home and then
paths.
like use my username.
So question, what the others are saying is that you're specifying the right files. The Slurm just doesn't know where these are located.
Kind of.
So it would be important to write the absolute file path so that Slurm, when it initiates these runs, knows always exactly where the input files are located. Because unless you are in the folder where that Illumina file is located, it wouldn't find that input file. So that, we can find this out though, or you could find this out if this really was the issue. Would you like, as the last step for your presentation, could you show us the output of that Slurm run?
Tail 20, the output.
For example, the last 20 the last 20 lines on it.
It's like it'd be like something like that. Like tail.
Tail space -n 20.
My only other thing would be he uses max time of 240. And I think
Yeah.
my group
And
used
then it's like-
360.
What is it?
So it could just be that
The N
small
dash N?
area.
Tail -n and then the number.
Yeah, so do like 20. That should work. Just give me a second.
Then of course you still have to specify what file you want, because right now you only did tail space -n 20, but what file are you trying to pull this from? Don't forget to specify the file too.